We're going to be looking at the convergence of three distinct scientific fields. The first is phage therapy, an older concept that uses viruses to target bacteria. The second is synthetic biology, which provides the tools to engineer biological systems. And the third, of course, is artificial intelligence. It's the combination of these three that is creating a new approach to one of our most significant global health challenges.
To understand the scale of that challenge, we can start with this number, 39 million. This is the projected number of deaths that will be caused by multi-drug-resistant bacterial infections, often called superbugs, within the next 25 years. This stark projection is what's driving the search for fundamentally new ways to treat infections.
So, this is the context for our discussion, the superbug crisis. It's formally known as antimicrobial resistance, or AMR, and it represents a growing global health emergency. Simply put, our current arsenal of antibiotics is losing its effectiveness, which makes it critical to develop new strategies for infections that we once considered simple to manage.
A potential solution now has been known for more than 100 years. We're now turning our attention to nature's own bacteria killers, a long-established principle that's now being re-examined with modern technology.
This brings us to the bacteriophage, or phage for short. These are viruses, and they've evolved over billions of years for one purpose, to hunt and destroy specific bacteria. In fact, they are the most numerous biological entity on the planet. And although they were discovered over a century ago, their application in a clinical setting has been quite limited, especially in Western medicine. That, however, is beginning to change.
So, the question is, why haven't these natural predators been a mainstay of medicine? Well, it really comes down to one central obstacle that has historically hindered phage therapy, and that's what we call the matching problem.
You see, phages are nothing like broad-spectrum antibiotics. They are specialists. A particular phage will usually only infect a very specific strain of bacteria. This means that if a patient has a dangerous infection, you can't just administer any phage. You have to find the single, precise key that is designed for that one specific bacterial lock.
This creates a fundamental conflict. On one side, you have the traditional way of finding a matching phage, which involves manually screening large collections of them. This process is labor-intensive, it's time-consuming, and there's no guarantee it will work. On the other side, you have the clinical reality of a patient with a life-threatening infection where speed and precision are paramount. The two just don't align.
This is where a new tool comes in. To solve this critical matching problem and really unlock the potential of phage therapy, researchers have begun applying a powerful computational approach, artificial intelligence.
In this context, AI is being used in two primary ways. First, it can act as a sort of high-speed matchmaker, predicting which phage will work against which bacterium. And second, it can be used as a genetic prospector, searching through vast amounts of DNA to find new and useful tools. Let's look at each of those in turn.
First up is precision matchmaking. The process works like this. Researchers can take the genetic sequence from a patient's bacterial infection and feed it into an AI model. That model, having been trained on huge data sets of known phage bacteria interactions, can then scan a library of phage genomes and predict, with a certain probability, which ones are likely to be effective. This can turn what would have been weeks of lab work into a computational task that might only take a few hours.
The second role is genetic prospecting. You have to realize there are countless phage sequences available in public databases, but many of them are completely uncharacterized. It's sometimes called genomic dark matter. Searching through this manually would be impossible. AI, however, can scan through millions of these phage genes to find the needles in the haystack, those specific genes that have very useful functions. So what exactly is the AI finding? It's identifying things like counter-defense genes, which phages use to get around a bacterium's own immune system. It's finding depolymerases. These are enzymes that can dissolve the protective biofilms that make many infections so hard to treat. And it's finding lysins, which are proteins that directly attack and rupture the bacterial cell wall. These are the kinds of high-value tools that AI is helping to uncover.
Now, just finding these useful genetic parts is really only the first step. The ultimate goal here isn't just to find better phages that already exist in nature, but to actually start designing and building them for very specific tasks.
And that process looks something like this. First, an AI system scans the databases and flags a gene with a desired function, like that potent biofilm-degrading enzyme we mentioned. Then, in the second step, scientists use synthetic biology techniques to take a well-understood phage, which acts as a sort of chassis, and they insert that new gene into its genome.
The result, in step 3, is an enhanced synthetic phage that has been custom-built for a specific therapeutic purpose.
This really represents the core paradigm shift that's happening in this field. It's a move away from being biological prospectors, just hoping to find the right tool for the job, and toward becoming biological engineers, who can actually design and build the precise tool they need. It is a shift from discovery to what's known as rational design, and that allows for more predictable and potent therapeutics.
Of course, as with any emerging science, there are still significant challenges. As this quote from the research indicates, the next major hurdle is about integrating data at different scales. Right now, AI is getting quite good at predicting interactions between different strains of bacteria, but it's less adept at predicting how a very tiny change within a single bacterium's DNA might alter the outcome. The key to creating truly robust predictive models will be combining that broad, strain-level data with this more fine-scale genetic data.
This all leaves us with a rather profound question to consider. We are witnessing the fusion of artificial intelligence with one of the most ancient biological systems on Earth. As these methods continue to be refined, it's worth asking if we are entering an era where our most advanced medicines will be designed by algorithms and then constructed from the genetic material of viruses. Based on the current trajectory of this research, that future is looking more and more plausible.